Neural network models are widely popular in fields like computer vision due to their unbeatable prediction accuracy. Yet, their performance on the prediction of biological phenotypes in genomic sequences remains modest and still under study. Users of neural networks face many choices. For example, we have to choose how to encode the input sequences as chimer vectors or simply converting the nucleotides ACGT as integer 3210. We also have to decide to use one-hot encoding or to embed the high-dimensional vectors into dense vectors via embedding layers like LSTM. Aside from the data encoding and embedding, we need to determine the NN model characteristics, CNN versus RNN, number of layers, dimension of layers, optimizers, embedding, and batch size. In this paper, we compare the prediction accuracy of different neural network models for different data encoding choices and different types of datasets, from small datasets with sequence length of 60 BP to datasets with longer sequences of 500 BP. Sample sizes varied from about 3,000 to around 269,000. Among some of the results, we found that adding more convolutional layers to CNN models tends to increase the overfitting, much higher prediction accuracy on training data compared to testing data. Overfitting is also more evident with larger datasets. Overall, we found the performance of CNNs deteriorated as a function of increased sample size, and these models suffered more from overfitting compared to other models.
We also found that the deep brain, which contains recurrent layers, outperformed all the other models in terms of prediction accuracy and robustness across datasets. The recurrent neural networks that contains LSTM layers show comparable performance to CNNs, especially when the LSTM layer was within the prediction neural network model, as opposed to having a separate autoencoder neural network for the sequences. Finally, despite the interpretability of doc2vec dictionaries, we found poor prediction accuracy when using them to encode the data across the dataset we studied. Lastly, we bring awareness to the importance of reproducibility in science. You can find all the code for our simulations and fitted models in the GitHub repository in the QR code. You can also read about our work in the BMC Bioinformatics paper. And this paper was developed by Zhaoyy Zhang and Songyang Cheng in Solis-Lemus-lab at Wisconsin Institute of Discovery in the University of Wisconsin-Madison. The project was founded by the Department of Energy. Thank you.